Okay, just copy and paste again for the people that are just joined. So I put in the chat links to add your contact information, but also a survey with some questions just to that will help the discussion. And they can just start the presentation. I just see them again. Some people have joined. So please see in the chat. There's the fill the contact information with the link, and also the you have a survey to fill that would give us a good idea on who you are, main interests, limitations of the eDNA time series, and critical gaps. You just have to copy and paste multiple times because the people that are just joined will not see the previous chat. Please fill that now, and then we'll do the presentation, and we'll go back on the questions and the answers. Okay. Do you see? Just see here.
It looks great.
See, yeah. Okay. Awesome. Okay. So thanks everyone for joining. That's great to have you here. So I hope this session will be a good opportunity to exchange on how we can define best practices related to field sampling. My concern is mainly about how can we develop an efficient sampling design without reinventing the wheel for each project, for each ecosystem. In my perspective, this is particularly important because many eDNA users are collecting samples because it's cheap and easy to collect, but sometimes I feel that there is less thinking and effort to develop appropriate sampling design for eDNA in contrast to direct species survey. I wonder how results can then be used to detect changes over time. I hope our discussion will help to target factors that needs to be taken into consideration for improving species detection, but also to improve our confidence on eDNA data interpretation when contrasting time series or data collected from various ecosystems. Okay, so in the last decade, we learned that eDNA can be released at different rates between species and environmental conditions, altering thus detection probability. eDNA then degrades, is transported, and diluted at different rates while interacting with the environment. All of those factors are playing a major role on eDNA detection probability. eDNA has first been presented as a magic tool where you take a scoop of sediment or water and up you can survey species and populations. Seeing all the complexity to develop strong eDNA sampling design, we invited scientists from various fields to study eDNA on itself. Strong fundamental science has now been done and available by ecologists, geneticists, but also by mathematicians, chemists, and others. Filling critical gaps to better define factors to be considered when designing sampling protocols. To develop best practices, we now have to link those discoveries to improve field, lab, and bioinformatics protocols. We know that eDNA has a different spatial and temporal scales than direct species surveys. That's what we have learned. Even when the lab method is considered mature and efficient. There's an increasing number of studies showing that eDNA varies seasonally due to both physical and biological factors. The good news is that eDNA can probably be used to study species biology, such as metabolism rate, biomass, reproduction periods, etc. But it also means that eDNA detection rate varies during sampling periods, ecosystem, and taxa. It also depicts the importance to adjust sampling effort to detect species at low abundance, that we need guidance. Factors to consider have been well described in the literature. However, they are typically broad and does not specifically mention how those considerations should be readjusted between taxa and ecosystem. In order to ensure eDNA data can be used by managers and that we can accumulate observations over time, we submitted a peer-reviewed report a few years ago, and I see there are people from DFO on the call, to basically provide guidance on eDNA that includes definition and consideration related to eDNA sampling, detection, and analysis for, and a reporting template as well that identified reporting requirement for eDNA services, service providers delivering results to managers. First, I personally found that we have been successful to highlight all potential variability that's need to be considered. The guidance were still broad and do not weight factors between taxa. For example, metabolisms might be important to be considered for fish or specific species, whereas reproduction period might be more important for invertebrates due to sporadic gametes released in the water column. So there are a major gap there. Recent reviews are still very broad and difficult to apply for a specific project and data interpretation. And the most important, from my perspective, is that we can learn from our errors and success and avoiding reinventing the wheel using artificial intelligence or meta-analysis if data, metadata, and protocols are all on a format that can be comparable. So better we compile sampling design, better we shall learn about efficient sampling effort, water depth, sampling periods, etc. for specific taxa and ecosystem. However, for the moment, since there's no general agreement on best practices to develop sampling design, I wonder how other groups are developing their protocols and which important gaps should be filled to move forward on best practices. So I was presenting now. I would like to do a roundtable to hear what is your best practices related to sampling design development in your program. Please present yourself, institution. I will put those questions in the chat to help people to provide an overview of their best practices. For the people that have not filled the survey, please do so. The links are in the chat. I guess I will have to copy and paste again. I see then some people have just joined. Stop there. Let's see. Okay, so I think we're back. Did I stop to share? Do you still see my screen or you don't?
No, we can't see it anymore.
No, okay. I will copy and paste again. Link here. Okay, so for the people that just joined in the chat, please fill contact information, but also there's a survey there to just help the discussion and any thoughts. So if you're okay for doing a roundtable, that would be great. And just as I said, present your institution. We'll put the questions that I think it might help to better understand your challenges. Oh, that makes it a bit funny, but there is basically 3 questions. How much sampling design is constrained by historical programs, so ongoing time series? Does your program aim to compare data over time? How do you define sampling design to meet the study objective? So how do you define number of sites, stations within a site, number of field sample replicate, time series, number of times that station and or sites are sampled? Do you consider a multiple condition? So I would like to hear from you guys how you do that when you start a new study within a new ecosystem. That would be great. So Katie, do you want to start?
Sure. Hi, my name is Katie Pitts. I work at the Monterey Bay Aquarium Research Institute with Francisco Chavez. And just thinking about we have a time series program that Francisco started quite a while ago, sampling 3 different stations in Monterey Bay monthly. And so definitely our eDNA sampling is kind of constrained to that program where we already have that ship going out. And this was kind of an added bunch of samples that we're taking on top of all these other kind of measurements that we've taken for a long time. But then we also have periodic kind of experiments where we go out and do concentrated sampling over much greater spatial scale and time frequency. And those experiments are driven more by specific questions we have that are with eDNA or the ecosystem. So we kind of have both going on where we're trying to take advantage of sampling that's happening and then also designing experiments that are specific to environmental DNA.
So some kind of 2 different clients, the people that are using for population survey and the people studying eDNA.
Right. So it's kind of 2 different pots of funding kind of too. We have like the time series funding, and then we have funding for these targeted experiments that have slightly different goals.
That's great. Thank you.
Yeah.
Raisa?
Hey, so my name is Raisa Maya. I'm one of the co-leads of the task group. And so in my case, I actually, in the work that I do, rather than going out and taking the samples myself, I work with the data that the observatories are producing anyhow and trying to integrate that, and then in the background to build a support structure and build the strategy to facilitate the integration of data coming out of the observatories. So the answer to your questions from my side is very much I use what's there. And it would be really great to hear from everybody else who's actually going out and taking the samples, what you're taking into consideration when doing that. Yeah, so very quick, yeah.
No, this is great. More meta-analysis, and that's the importance of combining data.
Yeah, definitely.
And Alison, what's?
Yeah, so we're working in sort of 2 different kinds of fields. One, much like Katie, we have projects where we're really adding eDNA into existing programs. So we're really constrained by what's already happened. We're just going to the sites that are already there. If I'm starting cold, if there's some question that comes up, like can we detect X invasive species or compare these aquatic systems, I think we start with the biology in those cases. We say, what are our species of interest? What do we know about them? When are they likely to be here? When is their spawning season? Where do they live? And then starting with that, and then we layer on access. Where can we actually sample? Where are they most likely to be that we can get to? Where can we not get to? So I think those are probably the two approaches. Stuck with what's there, and then starting with what we can do to maximize detection based on what we know about what we're looking for.
Shedding source.
Yeah.
Good, thanks. That's great. Brittany, answer. Brittany, answer. Do you want to present yourself and I can come back later. Chris Mayer.
Hey, Chris Meyer, I'm at the Smithsonian Natural History Museum. We do a whole suite of different environmental samples, everything from settlement plates to water samples to fish guts to just about anything. I think we're mostly interested in actually the library coverage and the distribution of diversity in space and time. So not so much from a monitoring side, but making sure that our biodiversity knowledge is sufficient to annotate these derived profiles.
Thanks. I hope I don't skip anyone. Kristen Litsky.
Hi, I think, does that work okay?
Yes, hopefully
Okay.
I said well the names.
Oh, no, that's fine. So we're mostly using eDNA for aquatic invasive species. And so we are just starting out with our program. And so we're really trying to figure out kind of how much sampling we should be doing. And very similar to what Allison was saying about, using the biology of the species as well as, when we can actually get to a location. The other thing that is kind of hampering us is that we're going to be sampling in some remote places in Alaska. And so we really need to have all of our ducks in a row before we head out there. And so some of our other issues are kind of how to handle the samples when we're in the middle of nowhere and, preserving them and that type of thing. Thanks.
Yeah, thanks. We have similar challenges in the Arctic of Canada for sure. Colleen Kellogg.
Hi, I'm Colleen. I'm in Vancouver, British Columbia, and I work in Canada. And I work for the Hakai Institute. We have a lot of different programs or projects within our kind of like genomics program that uses eDNA. Some are more like paired with oceanographic time series. They're a bit historical, but we've been sampling like eDNA samples since the start of it about 8 years ago. So those are sampled weekly to monthly. We also have more like community science programs where we have community members collect eDNA samples for us along the coast. And so as far as like picking those sites, we work with the community members to choose them. And then basically the number of sites we get depends on the number of kind of community members we can recruit to participate. And then we also have more like process-based or like discrete like kind of questions, maybe compare like using eDNA and then comparing that with visual observations of like kelp forests and stuff like that. And what do we see and how does that compare? And so then with those, with that study design, we often tailor that a little bit more to kind of like the questions we're trying to answer more specifically, whether it be methodologically or so we have a lot of different things going on. So it kind of depends. As far as like field sample replicates and stuff, some of our samples that we collect or like our kind of more oceanographic time series are often pretty water limited. So we don't get to do replication because like every drop of water that's brought up is precious for all the different people sampling. But for like our community science program, we do a lot more kind of replication and like sampling blanks and stuff like that to control for any sort of contamination that might be introduced in those things. So, yep.
I have a quick question for the weekly and monthly survey. It's mainly to learn. When is the optimal period?
So like basically we've been sampling weekly because we're like, well, for me personally, I work a lot with like prokaryotes. And so we're kind of trying to sample at the time scale at which their populations can turn over and change. And then we layer on to that using other markers to look at other planktonic groups like zooplankton and phytoplankton. And so with those, we are starting to look into like how often do we have to sample to actually like resolve the community. Yeah, so that's, and basically we've been sampling that kind of, that's a bit dictated by the like oceanographers that are going out because they want to sample on the weekly time scale. So do we. But we sample like different, we don't always sample for like all of our eDNA suite. Like sometimes we do eRNA and that's only done like bi-weekly and stuff like that. So.
Yeah, thanks. I said that because it's mainly a monthly survey for me, but we're trying to move to more weekly to really understand the temporal variations. So.
Yeah, I guess like kind of what I've started to realize even in like the microbial side of the data set is like there's also certain times of year where it may be more, well, not started to realize, I kind of knew it intuitively, but now we're seeing it in the data, like maybe in the spring sampling weekly is really important, but in the winter things are a lot more stable. And so hopefully we can kind of resolve that and potentially adjust sampling accordingly so that the field teams don't die of exhaustion. So, yeah.
And do you think during winter it's related to volatile conditions or more the species biology? I'm saying that because we're contrasting south to the Arctic, and we first thought would be really stable, and I think we're seeing like a lot of variation. So
yeah, and I guess it also depends on like how much you like dive into the like prokaryotic groups, for example. Some are more stable and some are changing more. I think it in part has to do with the oceanography, like the physical conditions, like the nutrient levels are much more stable, but I would say wind mixing is a lot less, is a lot crazier in the winter. And so that is also causing some differences. Yeah, so things we're still trying to work up and publish on.
no, that's great. In the Arctic, we're looking for the ice cover to see the effects. So,
Oh, yeah.
thank you very much.
Mhm.
Philippa Martins. Philippe.
Hi.
Hello.
Sorry for the delay. I just entered. Can you just make an introduction? What do you expect me to say?
Sure, I will. So there's if you can introduce yourself, institution, but also if you can answer questions related to how you define your sampling design. If you are someone's collecting samples, how does your program aim to compare data over time, and how your sampling designs can affect that? And yeah, any guidance about how when you start a new survey, how do you define the sampling effort and sampling period? That needs to be
Okay.
That's a big question.
So I'm Philippa Martins from the Biopolis CBU in Portugal. I confess that I mostly work with freshwater biodiversity, but I'm starting a project with rocky intertidal algae. So this is why I was interested to know in the marine part what have been done and what are your experience with this environment. But mostly my work is, well, we start with a pilot study. So basically at the moment, we don't have a temporal study. So we don't have samples across time or across different regions. So we always start with a pilot study with, at the moment, we have 6 beaches along a climatic variation, I can say. And we try to do biological replicates in the beaches, at the 4 in each beach. So yeah, so basically we start small with few samples, but try to make effort with replication, biological replication. And what was the other question?
I think it's good.
So,
yeah, basically, you try your best to deploy as much as you can, and then you will learn from that.
Yeah, exactly. Yeah.
Some people,
and because the communities that are not, well, that there's not the reference collections are not enough, are not sufficient to do the meta. So we use meta barcoding. So we are also collecting specimens and identify species level. We try and then barcode and then create our own reference databases. Yeah.
This is great. Thank you so much. I put in the chat for people that have just joined recently. There's the notes where you can fill your contact information, but there's also a survey where we can see an overview of the people on the call and
okay,
how to move forward to have the discussion after.
Okay. Thank you.
Yeah, thanks. I don't know if I say it well, Lulia Sinti.
Hello, Yeah, I'm Julia Sandi. I'm working at the European Marine Biological Resource Center, and I'm actually working on a genomic observatory. So we're using eDNA to capture the metazoa communities that are in the sea bottom, both from soft sediments and hard substrates. And we are also targeting different microbial communities in the sea, in the water column and the sediment. And yes, so for us, the sampling design does include marine stations with rich sampling history that are, let's say, historical marine biology sites. And the project was basically built on those long-term time series stations. And then it was complemented with other stations that recently initiated time series, biological time series. Yes, and one reason for that was because we eventually want to compare samples with past data, and also because those long-term stations usually have very high experience in those regular sampling campaigns, and also in the different methodologies used. So for example, we have stations that are doing metabarcoding, regular metabarcoding samplings since 2010. So they have a really rich molecular biology time series from their area.
Yeah, that's good. You're lucky. There's a lot of, it seems to have a lot of data to base in sampling design. So. Thanks a lot. Jessica Rader.
So hello, I'm at the University of Bern in Switzerland, and my main focus is studying microbial communities in recirculating aquaculture systems. And there I look at freshwater and saltwater systems. And yeah, basically just trying to understand how these are affecting the systems themselves and the fish health. So I'm not actually in the field. So my limiting factor was how often could I drive around Switzerland collecting samples before I was exhausted. And my other interest is more into eDNA tool development, for example, to develop dried qPCR assays that can be then moved to sampling machines or being combined with lateral flow strips. So that's all.
Yeah, thank you, Jessica. Trying to see the names as Flipperain, Jody Van de Kamp.
Hi, Anais. I joined the meeting late. Are we just giving a quick introduction or?
A quick introduction, but also if you have any strategy when you start a eDNA survey about sampling design, how you develop a new sampling design, what's your best practices to develop sampling design and time series data. So basically, an optimal detection period and sampling effort.
Yeah.
If you have any recommendations or what is your own best practices?
So I think it's what Katie has brought up before, historical, really. I lead the IMOS Marine Microbiome Facility, and we take time series measurements from 7 reference stations around Australia. We've been taking microbiome measurements since about 2010 or 11, but those stations have been going for a long time before that, and so they already had a sampling campaign designed, and we started out by piggybacking on that before becoming a formalized data stream. And one of the good parts about that is the fact that we get all the other measurements that come with it. We get all the phytoplankton and zooplankton data. We get all the physical chemistry data and everything else. Those programs themselves are constrained by logistics and budget. How much money have we got to get out there? How many times a year? Generally, it's monthly. But again, because this is out on the shelf, these time series stations, weather plays a part in it. Obviously, boat logistics can sometimes play a part in it, but not so much. Time to station. So they need to be full day sampling trips for a lot of them because it takes a long time to get out on station. Yeah, I think that would be mostly it. And then I also am involved in some coastal time series data sets, which are right on the coast or estuarine. And these ones also piggyback on existing monitoring programs. So we're sort of beholden to when those programs go out.
Yeah, that's great. I think most of my study, I do microbiome, but most of my study are water or macroinvertebrates, so we don't have historical data. That would be a dream.
Yeah, yeah.
I think that's a great point that, you know, so often we're saying we're designing our eDNA studies based off of data we already know about the system. So yeah, if you're piggybacking off of sampling that's already been happening, you might already know a lot about the system that helps you decide what you need to do on the eDNA side as well.
Kelly Goodwin.
Hello, can you see me?
Yep, I do. Okay.
So I would say in general, most of our sampling is tied to traditional sampling because we're piggybacking. I think probably more importantly is do we know, let me back up. So all of those samplings are I did your survey. I mean, they're all some sort of biological thought went into these surveys. I mean, some of them are 40, 50 years old. They're long-term dedicated operations, and thought went into them. Whether that logic for trawls or whatnot or chemical oceanography are the correct match for eDNA concentrations, I would say we don't know. And it wasn't until we were talking about this, I was like, well, of course there was biological thought that went into our surveys, but wait, but they were still based on what was known for traditional, give or take, so, and primarily the idea of when we think about fate and transport of water containing an eDNA signal, most of these surveys are fixed spatial. None of them are synoptic, like following a water body, because no one designed things like that. So that's an interesting thing to think about, and how would we approach the science of whether these surveys are correctly suited for eDNA. I'm not sure, but I'm intrigued to try to figure that out.
Yeah, I think there's two questions. There's really in terms of sampling design. So that's great. If we can piggybacking, it's well limited by funding. So there is an advantage to do that. You can do more often, or you can have more samples. But personally, I do wonder if data can be comparable over time. If you do that once a year, because you're piggybacking on species survey, and then if you see a changes 10 years after, can you really say something? I'm not sure. It might just represent the species reproduce better because of global warming or other factors. So that's basically I think there's really two questions around that.
Mm-hmm.
I think that's a good point of, sorry, I would like, how do we evaluate how well these surveys are suited for environmental DNA? I think you're right, Kelly, that a lot of surveys were designed maybe to catch something specific or the best opportunity to catch what they wanted to catch. So in eDNA, what's the best opportunity for us detecting what we want to detect? Or if it's like a whole community, when do we get the representation of that whole community? They're interesting questions.
And I personally go as often as possible, which doesn't make any sense at some point. At some point, I should have more biological and rational, even if I know them. It's often there's a lot of uncertainty right now in most of the surveys. So.
Yeah, and in the survey, you had a thing that was like maximum user. There were some things I clicked in the survey, which isn't true because we're really selling eDNA to the agency to try to reduce the current burden of sampling, you know, saying that it'll be easier than things like trawling or whatever, you know, to be able to go more often, especially if we're using autonomous sampling somehow, or we could get out with a small boat and not need a large white ship. So to a large extent, it's economic value. It's all about the, you know, trying to be able to meet our mission, meet our observational requirements, spending less money.
But that will be cheaper in the future. Automated samper, I mean, they are coming very quickly. There are many around and developing and things like that. So eventually, might be very effective. Or maybe once a year, it's fine for some species too.
I think Nick, sorry, you have your hand up, but I just want to say too that, you know, yeah, it is a balance between with eDNA, maybe we can take a lot more samples as compared to trawl measurements, but yeah, trying to figure out what's the right mix. Nick, I don't know if you want to jump in right now.
Sure, I can do that. Sorry, I don't mean to derail the current conversation, so feel free to go back to that, but I was going to chime in with my own experience. I'm a research scientist with an eDNA company in the US called Jonah Ventures, and we work with a variety of clients from marine to freshwater to diet with metabarcoding and qPCR. And often our clients are following their own sampling strategies, but we are working on one project that's starting or continuing and ongoing with fieldwork starting super soon, AKA tomorrow. And this project's with Nature Conservancy and Oceankind to ask some basic sampling design questions for eDNA and biogeographic questions in general. So mainly about spatial temporal extent for capturing patterns of richness, species occupancy, and how well eDNA can do that given known diversity of a system or estimated diversity of a system. So we started with a kind of a proportional stratified random sampling design by habitat type in this atoll region. And we opted for that instead of a more uniform sampling per habitat. So that's an interesting question in of itself. Do you proportion your sampling by target habitats? And then we prioritize spatial coverage over site replication for our spatial questions. So instead of sampling 100 sites 10 times, we're doing 1,000 sites one time sort of thing. That general framework. And that's looking at the spatial coverage. And then we're also looking into other questions about appropriate distance among samples, species accumulation by sample number and replication, and then diversity estimates between top bottom samples, surface water versus at the benthos, or along regular depth intervals to try to build out that curve. So it's basic, asking some basic questions while also trying to characterize the diversity of a system.
Are you going after kind of whole community biodiversity or certain like targets, groups of organisms? I'm just thinking about fish versus like invertebrates or, phytoplankton. Are you going after it all, or you do have like certain needs that you feel, or certain communities that you want to go after most?
Yeah, ambitiously, we're targeting all of it. So we'll do, for metabarcoding, we'll look at fish and then inverts and then algae as well. And we really just have some clear knowledge of the fish communities or, kind of fish diversity. And so we can relate that back to the kind of the morpho and previous surveys of, catch and visual and acoustic surveys of what we know should be there for fish, but we don't really have a great idea of that for phytoplankton, for instance. So eDNA will kind of be the baseline in that situation. And then we'll look at diversity estimates regardless of who's there, internal to the database itself, the eDNA database, that is.
Yeah, that's great. I feel personally that's some kind of the best approaches, but what I wonder is how we can learn from other studies, and when it's done very extensively like that, how we can apply it to our ecosystem. And this is where it starts to be really really difficult to differentiate between taxa, ecosystem, and different different environments and limitation. Logistic limitation. Thanks. I think I lost track of who. Luke Thompson, you want to present yourself?
Yeah, let me see if I get my camera working. There we go. Yeah, so I work with Kelly and Nastasia. I think the way I would answer this question about sample design and historical programs. So one of the, so I'm at NOAA in Miami, and we have a nascent collaboration with the Ghost Ship effort. We also have an MBON in South Florida. Both of these are ocean-going vessels that have long-standing sampling programs, and especially on the ocean, it's a challenge to get water from the CTD because there's so many other activities that have priority because they're the main motivation for the crews. So I guess I would say, you know, the study design isn't always exactly what we want it to be. And so thinking about how we can piggyback on existing field operations and historical programs, we want to try to maximize, you know, the science we can do with those restrictions. So like, for example, if possible, we'd like to get a deep cast, you know, every few stations where we can do eDNA from deeper depths. But oftentimes we have to settle for surface water from, you know, a flow through system if we feel it's you know, not contaminated by biofilms or whatever. We can do that. So I guess that's sort of the other side of the coin, is kind of like what can you get given the restrictions that you have.
So what is the most missing part? Let's say, which information will be best to
yeah, so I mean,
guidance?
the missing part physically is the deeper samples. It's harder to get deeper samples because it takes more time to you know, on an ocean vessel to do a CTD cast to 1,000 meters or 500 meters takes a sizable amount of time, and then, you know, multiplied by however many stations, and then there's only so many days at sea, and there can be delays and so forth. So oftentimes, you know, so that's a limitation. So if there's ways, you know, technical ways to get around that, you know, with drones or that sort of thing, AUVs potentially. And then I guess, you know, another point, you know, with these kind of working on existing sampling programs is to, you know, ideally get the other environmental data that you want or need. So like, for example, on more physical or chemical oceanography cruises, they might not take all the biological parameters that you would like sometimes chlorophyll is not measured, or fluorescence isn't measured as a proxy for chlorophyll. So yeah, that's what I would say about that.
Yeah, that's great. Thanks. You're working on much deeper environment. Here we are, I mean, Basin Bay of Fundy, so it's really mixed and not so deep.
Yeah,
so it's very different.
Sure.
And we wonder how much the depth change between, you know, like well, in the Gulf region in Canada, it's 6 meter depth deep, so it's quite mixed. It's not a big challenge. It's very different when you go offshore and some locations. So how much it's comparable if you are talking about like if you want to use about eDNA in terms of detection rates between different ecosystems, it's starting to be very important.
Yeah, and the other problem with these open ocean stuff is often the biomass is much lower, so you need more water to get DNA, and so that complicates everything. All the things I mentioned about like competing for water becomes a bigger issue, because instead of maybe being able to get by with 1 or 2 liters, you need 8 liters, say.
This discussion around kind of having to make adjustments based off of which like historical sampling program you're piggybacking on, it kind of reminds me of our discussion around sampling of remote systems, or like some way to better indicate when you have to make adjustments, or like what are the most important considerations, I guess, when altering protocols or altering kind of, yeah, your sampling program. We've talked about that, I think, in terms of where you're sampling really affecting, but this is my first time thinking about it in terms of kind of when you have to piggyback on another cruise, the kind of adjustments that you have to make.
We have discussed that in the last 2 days, but one factor very important is that most people will not publish when they're not successful. So basically, if we don't publish when we're not successful or don't report our data somewhere, we will definitely reinvent the wheel. Like, let's say you don't grab sharks in the surface, or the opposite, or something like that. You would just take a couple of years before you might be successful. So that's some things I found that often the protocols are not easily accessible, or they can vary between sampling or something like that, how they are reported and how we can compare that. So it's difficult to learn from other surveys. So I think all of the effort of OBPS to make sure that all data are provided, including non-detection, that's awesome. Hey, thank you very much, Luke. Martin, I'm very bad to read names. I'm so sorry. I have a very complicated name for English speakers. So my full apologies.
Hi, Anahita. Martin de Brouwer, but Martin is fine. So I work for CSIRO in Australia, which is the Australian National Science Organization. In my current role, I don't actually do eDNA projects myself. I've got more a role where I'm trying to mesh together the needs of marine park management with the potential of eDNA. So what I'm trying to create is sort of a roadmap on how to best integrate eDNA methods in their monitoring program. So looking from that point of view, experimental design and all the sampling questions are massive, because the system in Australia is quite interesting, because we've got federal, I think, national marine parks, which are usually offshore. So they range from, we've got deep sea canyons over 6 kilometers deep, but there's also coral atolls that essentially are just surface water. But then you also have state waters, which are heavily used by recreational users with fisheries. So there's a whole lot of stakeholders. And depending on where you go, there's different existing monitoring programs. And so the question that I often get from marine park managers about the design, it's usually a budget thing. There's either you get the people who are very interested to use it, but for them it's purely a logistic question, like how can we fit this in what we have already and with the budget that we have already, which is usually quite minimal, their budget. So they tend to work with whoever researchers, whichever like universities that are nearby that would be interested in doing eDNA work. So that's one of the questions, and something that's still a question in quite a lot of resource managers is they literally, I've had people still tell me not too long ago that they do not believe in eDNA. So, which is, I mean, there's still quite a high level of skepticism, especially if people are used to traditional methods. This is for management, right? Like the eDNA research community over here is fantastic and very collegial, and there's very, very good research happening, but it doesn't always flow through to the end users. And so one of the things, I've mentioned this 2 days ago, we created with the Australian New Zealand eDNA Society, a big group of researchers, we created best practice guidelines, which are not standard. They're really how how would you develop your SOP? How would you develop a standard? What do you want to think about? Because those guidelines are not only marine, they're for terrestrial biosecurity, they're for aquatic, they're for pretty much anything. And so we found in those workshops that when you're thinking from the end user's perspective, more often than not, you have to actually tell people that they have to have an experimental design, which was quite surprising to a lot of people. So that's one of the big points in the guidelines, where we have to start with, like, you actually, eDNA is not going to solve everything. Like, it's not suddenly magically you can just take some water and you'll know everything. You still have to have a design. And so I'm just looking at it now. In the guidelines, there's a whole lot of guiding questions we set up if you want to design your study with eDNA, and they go from a lot of it, what is the goal of your survey? Because an end user might have the goal, as people have mentioned, they might want to detect endangered species or invasive pest species. But then for our Commonwealth or federal, marine parks, there's a lot of unstudied areas. So there's general baseline surveys in deep sea canyon or in new marine parks where they don't know. So there's pure exploration and baseline monitoring going on. And then for the state parks, a big goal there would be to look at changes in the system. So they'll have, in some parts, long-running historical data sets. So then you'd have to calibrate it. So it really depends on the question of the end user, and that's what we kind of set as, and we repeat it quite a few times because it seems to be forgotten, as we've seen in a few projects. So look at what your study goal is, what do you want to detect, and then it's logistics. What have you got available? What are your boats that you've got? What are the tools? What is the in-house expertise that you've got? Specific targets of like taxa that you're targeting. What else have we got in there? Which characteristics, if known, of your organism might affect sampling methods, whether there's spawning events for certain species that might have an effect, whether there's a larval versus a sedentary state. What else have we got? It just kind of goes on. There's a whole heap of guiding questions we got in there, but yeah, we've structured those questions really about what do you want to get out of it, because the guidelines we provided are more registered for a practical use than pure research. Having said that, we do highlight that there's the ecology of eDNA needs to be studied better, because there are definitely questions that I get from stakeholders, from resource managers, where they are, but just how far can we detect? And what about temperate waters in Australia? They tend to have lower biodiversity versus the coral reefs. We've got this very high biodiversity. What's the difference there? So those questions are still they're definitely asked, but they should be taken into account when designing. In this case, what I'm talking about is monitoring projects, government monitoring projects. So different than what some other people are talking about. But yeah, the whole list can be. I'll share the link again of where people can find the guidelines. It's the guidelines are in there. I'll just share them. So that's about it.
No, that's great. I mean, when people don't believe it, I mean, the reality is indirect methods are always there's always a risk. It's indirect, so I mean, it's there's always a risk. But if you are seeing that often and you test your lab and your method is efficient, a presence is a really good flag to think that the species is there. When we don't detect eDNA, that's quite true. Maybe the species is there. I will not trust an absence yet in most of the survey. Whereas a presence, I can usually trust it. However, the absence can be very costly if you just try to survey randomly with this, at the wrong depth. So that's the same. Very expensive.
That's the same for the methods that are currently used, like the video methods that are currently used often in the marine parks here. If a fish doesn't happen to swim by, you don't know if an absence is a true absence either, right? So it's not necessarily unique to eDNA that the absence, whether or not that's true. One thing actually that you reminded me of that is a big question It's actually two things. Management always asks for abundance. There's no way around that question will always come back from resource managers. And it's really hard to get people to think in different ways about abundance, whether it's through different kinds of modeling or through different kinds of repeated sampling. So it's quite a hard question, and I don't think we need to be able to answer that straight away, because there's a growing body of research. It's some really good review papers that show, especially for meta barcoding I'm talking about. But it's something that will always come back as a question, the abundance question. And I think kind of linked to that, which what I get from some of the current monitoring programs which use traditional methods. And that links to one of the questions in your survey. I think it's question 3 in your survey. The main motivation and question C is replace traditional methods. I'm not sure for where this survey was designed, but in Australia, it's actually quite a laden question, because what I feel in people doing traditional monitoring, they're quite comfortable with their method, and sometimes the monitoring is done by the experts who developed a certain traditional method, and they feel threatened by eDNA because there's so many things that can do, and no matter how often you say it's complementary, I think at some level they see that it's cheaper or there's benefits that they can't offer, and I've had kind of defensive reactions from people doing traditional methods that think exactly that what we're trying to do in the eDNA Society is replace everything they are trying to do. And I think in our communication, we should probably try and it's complementary. I mean, there's some old-fashioned methods in a marine environment that should go. I'm thinking like rotenone collections where you kill everything. That probably hasn't got much place. But I think we should try and emphasize that it's not a replacement. It's a complementary method to just get more information about the environment that is of use to better conserve or to whatever the goal is, right? Because it will help being people getting more collegial. So I understand where it comes from in your survey.
Well, yeah, I did the questions, and they helped with a lot of people from the call, and yeah, it's basically always surprising me because in my perspective, we survey so tiny, and when we do the eDNA, like the eDNA as a powerful to survey a much larger scale, and whereas a specimen survey, you provide a lot more information, but most of the time you're killing it, or you have like negative impact on the environment, or so the reality is, in my perspective, eDNA is very different time scale, and we should adjust. But that's my perspective. But if we can survey the full Arctic or the full Atlantic Ocean, that's very powerful. So that's
I completely agree though. Like a lot of things can be replaced, but I'm trying to say this respectful, but let's say the entrenched experts on certain traditional methods don't they feel it can feel threatening if you can go, well, you might be able to survey a 50-meter transect with your ROV, but we can survey, as you say, the entire Arctic. I can see why there's a bit of hesitancy from their point.
Yeah.
But it is super powerful, even with the limited understanding.
It's powerful for general area at the scale. However, you don't get help of the animals, you don't have the size, you don't know, like there's so many other questions that is not filled.
Exactly. Absolutely.
Yeah, good. Thank you. We'll definitely see the guidelines.
I'll share them.
Yeah, so that'll be great. Thanks. Melissa Morrison. Hello, Melissa.
Hi, I'm Melissa. I'm with Fisheries and Oceans Canada, and I was determining biological and physical factors that affect eDNA when relating to rare species abundance in a river. And so The sites and the timing were chosen based on species life history and also timing around the conventional monitoring. And so it was also important to have frequent sampling. So depending on what time of year, if we're trying to monitor during migration, then we would want more frequent sampling so that we can capture the environmental variation in a season. Limiting factor was discharge. So in a river, if we were able to get discharge, unfortunately, this river was extremely difficult just based on it. Yeah, it's a really windy and has crazy depth variation. So yeah, that would be the limiting factor. That was the most difficult to get. That might have been like one of the most informative pieces of information, especially when we're sampling, say, in the spring versus in the summer, when there are such dramatic stream flow differences. Yeah.
Yeah, thanks.
I don't, that just makes me think of, I don't have to sample in rivers, but I'm wondering how like traditional sampling efforts account for differences in the river seasonally, or I'm wondering how how do the other methods try to account for those differences?
Yeah, so one of the main, so during the summer, it was Atlantic salmon. And so they did electrofishing just in a 100 square meter sites. And so that was, yeah, that was good for that. We would able to know an estimate of abundance kind of throughout the whole river, but still we didn't know exactly where the par were at throughout. So our sampling tried to account for, like we sampled around the electrofishing sites, but we still weren't sure where they actually were at the time of sampling because they could move around. And then during the spring, we sampled during the smolt migration, and they had a rotary screw trap, and so they caught a percentage. And so again, we sampled upstream and downstream of that trap.
And there was a pretty good relationship if it's corrected by temperature. So if you,
yeah, temperature. And then I'm looking at data now that captured from other people that they didn't get temperature, and so then we don't have that piece of information. Yeah.
Yeah, it's a lot of. I work with Melissa, and this is very variable, but we see a pattern. It's just how to learn about all this pattern. Yeah, I have a lot of questions still. Thank you very much, Melissa. Miguel Weiss. Michael Weiss. Or I can go Nastasia Patton. Do you want to present yourself?
Hi, I work with Kelly and Luke both at NOAA as well. And so I think they covered a lot of the logistics and challenges that we think about when we do our eDNA sampling. I think maybe the one thing I would add is, despite all the challenges that come with trying to add on eDNA to a more traditional survey method, which there are many, but there are also advantages that I think, coming from like a more of a pure microbial background, you know, when I was doing just regular microbiome sampling and sequencing, we would not be getting the types of metadata that we're getting from these traditional surveys. So like fisheries trawls and, you know, marine mammal and bird observations, and a lot of, yeah, elements of these surveys that are actually really valuable and wouldn't, coming from, yeah, microbial oceanographic background, wouldn't necessarily include them. So I think, yeah, I think it's just worth thinking about the all the positive enhancements that can come with combining survey methods, even if they can be logistically quite challenging, not just in the field, but also like when it comes to the data analysis side of things. That's just been a massive challenge, because different scientists collect data in different ways and record it in different ways, and they don't all necessarily talk to each other. The data don't all talk to each other very well. So yeah, that's probably the one thing I would add to what Luke and Kelly mentioned about our NOAA programs.
And to ensure all of the data have been collected and which data, guidance on which data should be recorded. It's very important. Minimal reporting and how it has been collected. Thank you. Nelly Gagné.
Hi, I'm with Fisheries and Oceans Canada. I work with Anais on some projects. So I'm going to talk about something that we didn't work together on a couple projects. And because there's been a lot of things, interesting things said, I'm going to talk of the takeaway message that I want to give you about two of the projects we've been working on. One is we were looking at a population of fish, striped bass, in a river. That river is influenced by the tide, so it's a tidal river near the estuary, and the population is quite big. We were talking of hundreds of thousands of fish, and we were trying to see if we would eventually be able to evaluate the size of that population using eDNA, because these fish, they overwinter in a relatively short section of the river. We're talking of about 10-15 kilometers long, and the river is not that wide. So what we did 3 years in a row, over the ice, we created a grid pattern, and with holes, we were able to get the water. And what was amazing, and that's my message, is that when you look at the concentration of eDNA that we could get out of each of these transect and holes in all the transect, you kind of get a heat map of the distribution of the fish. And the heat map that we created matches what they could see with radars and other methods to try to determine where the fish were. Except that because it's a river and it was submitted to tides, that heat map is kind of shifting left and right with the tides, if you follow me. So you have the exact distribution of the population with more concentration of DNA where there's more fish from the other measurements that were made. But because of the river that is flowing, that eDNA, these eDNA concentration do represent the fish, but it's just moving left and right with the tides. So it was really amazing, and it was repeatable 3 years in a row. So one of our first big projects tried to determine if there's a possibility to evaluate the whole population of fish. I think it's promising. We're not done yet because we need to have a standard curve of the fish biomass, the eDNA that is shed, knowing the fish biomass in a known volume of water. And it's kind of difficult to do at the temperature where we were doing this measurement, because we were working in the winter, but when we work in the fish, usually you're not doing that at 2 degrees in a hatchery. So it's kind of where we're stuck with our evaluation. But anyway, I thought it was quite interesting that the distribution was so precise. The water wasn't, it's a river, so there's mixing, but the heat map was so clearly matching where the fish are located that it was quite interesting to see this. The other project that is ongoing, it's been 3 years now of sampling. We sample water alongside the stock assessment that is done for for our evaluation of fish stocks in our area. And this is also quite, I find interesting, the fact that some of the sites, the station, are not so far apart, and our sampling is either bottom because we want to catch with the metabarcoding, we want to detect the benthic fish, for example. And so we have two samples, bottom and 15, 25 meters below the surface. And even where it's not super shallow, we can see a big difference between the metabarcoding signal from the bottom and the top sample that we take. So the eDNA, my message is that the eDNA, which is something we were not sure about initially, the eDNA is going to be detected, observed where the fish are. That's what we want to say. We also did some cage experiments where we were removing the cage from the environment and look at how long the signal would be detectable, and it's not staying long. Like the eDNA disperses is not detected quickly after the fish are removed from where they were. So I think it's information that needs to be, the more we get that type of information, the more informed we're going to be about our sampling designs. This is a lot of the things we do. We try to understand more how the eDNA works, but we need a lot more of that type of research going so that we can better design all our research projects in the future.
Yeah, I was thinking about that when you have to design for species, rare species. Nelly and I, we have a project on sharks and tuna and large organisms, which are, I was more used to it with small and zooplankton or abundant species. So we don't detect them often. We are detecting them sometime, and clearly the shark was not there, and we collected a bottle of water. So there's still a chance, even if the organisms is not right there, but clearly the persistence the signal doesn't stay long. And when they are migrating after in the south, we're definitely losing it. So we have a window, but that window might be shorter than what we believe first. Thanks, Nelly, that's great. And we have a lot of discussion, Nelly and I. It's very difficult to make sure we are confident on something designed for the first few years for each project. Nick Jeffrey.
Yeah, hi everyone, I'm Nick Jeffrey, Fisheries and Oceans Canada, based in Nova Scotia in Canada. My group focuses on marine protected area monitoring and design of future conservation areas as well. So we use a mix of population genetics and also eDNA, primarily metabarcoding, for monitoring both fish and invertebrates in our marine protected areas in our bioregion anyway. So we've done a few kind of small scale pilot studies in the past, and then over the last 3 years, we've been doing some eDNA sampling in a couple of places called the Eastern Shore Islands and St. Anne's Bank Marine Protected Area. And that's mostly limited by vessel access in the summer. So we're down a couple of science boats on the Atlantic Canada, and we share a couple of science vessels with another DFO region. So we typically get about 2 weeks at sea, and we typically just sample along transects from the near shore to deeper water within marine protected area, and typically sampling surface and bottom samples to try and capture as much diversity as we can. And so we've done that for 3 years in a row now. So we're just starting to kind of look at how the signals have changed with time for a couple of years, and if we're getting kind of any repeatable results or anything like that. And we just want to echo a couple of the other issues that have been brought up by Luke and our others here. How we are would like to get water samples from some of our more regular surveys, like our Atlantic zone monitoring for oceanography, as well as our summer and winter trawl surveys for fisheries stock assessments and things, but it's hard to be able to claim any of the water from their rosette samplers, or have someone on the boat to do the filtering, or ask them to do the work for us. And sometimes they'll collect water and throw it in a bottle and freeze it for us, but we don't have as much control over contamination, and it's not as ideal as fresh sampling either. So but we would like to take advantage of regular surveys, particularly to get to some of our deeper conservation areas that I myself don't regularly get to. So mostly limited by vessel time right now. I think that's it for my group.
You have a very large spatial scale, so in your study, which is
yeah,
it'll be very interesting because you have a lot of replication for a certain level, certain scale.
That's right. Yeah. And our marine protected areas here in Atlantic Canada anyway we're trying to make the 25% protected ocean by 2025 targets. And so we have, thousands and thousands of square kilometers of area to monitor. eDNA is one tool of our monitoring program for the MPAs, but it is a pretty primary tool for us because of its ease of sampling and relative ease of kind of processing the results instead of things like terabytes of video and so on. But yeah, we do have a lot of a big spatial scale, and we also do work with other Atlantic DFO regions on their similarly large MPAs.
Great, thanks, Nick. Looking forward to see 3 years in a row. That would be great.
Yeah.
Name has all switched in the sorry, let's track. Who would like to jump in and present themselves? Sarah Stinson. We don't hear you. Are you mute? No, we don't hear you. So bad. Maybe you can. Okay. Sorry about that. Feel free to join me by email or write your some text in the chat. Can you? No, it doesn't work still. Okay. Jonathan, ready?
Oh yeah.
Did that work?
Oh
no.
Yeah, that works. Yeah. So Jonathan, you will be next. Sarah, do you want to go first? Sarah Stinson, you can go. Okay, let's go, Jonathan. Ready? The floor is yours.
Sorry. Oh, did that work?
Yeah, now it works.
Oh,
okay, go
sorry.
ahead, go ahead, Sarah.
I think my headphones are not compatible with my monitor setup. Sorry about the delay. I'm Sarah Stinson. I work with the California Department of Water Resources, and we are incorporating eDNA into several different projects that we're working on. We just established a new genetic monitoring laboratory for the department. And right now our projects are focused on single species assessments, looking at endangered or threatened species. But we're planning to expand in the future to do some metabarcoding projects and other projects as well. But the main project that I'm working on right now is looking at improving the monitoring efforts for an endangered fish in California called the Delta smelt. It's hard to monitor, it's very rare. And so we're exploring both single species qPCR and Sherlock CRISPR Cas13 assays to look at this species. Some of the limitations in our system are it's very rare, it's a large delta, it's an estuarine system, so we have tidal influence, different salinities, turbidity is a big issue in our system. And the ultimate goal is to use eDNA as a complementary sampling tool with the existing monitoring programs that we have. But there's so much we don't know about our detection probabilities that I'm developing some pilot projects to try and nail down how to optimize our sampling. So looking at different sampling approaches, large volumes of water filtered through a townet, for example, versus like an eDNA backpack setup that we could use on a boat that's already out doing townet sampling. And we're also doing some caged studies where we'll have hatchery bred Delta smelt present upstream from where we'll sample, so we can understand a little bit better about the shedding rate and some of the environmental factors and how that's going to affect our detection probability, with being able to say, yes, we can detect them, or if we can't detect them, what is the probability that they're actually absent or that we have missed them somehow. So that is one of the projects I'm working on. And yeah, just a lot of limitations with, like other people here have said, with trying to coordinate with other sampling efforts.
So the guidance that will be helpful for you to have to start will be dispersion and turbidity effect.
Yeah, I would say those are two main ones. Also, we're looking at different life stages of Delta smelt and what the shedding rate might be between life stages, because that is unknown at this point. I think there was a presentation yesterday from a local, from the California EPA had a presentation. They were talking about detecting a large single-celled organism, and it turns out it just doesn't shed at all because it's asexual. There's just nothing in the water for anyone to be able to detect. So we're trying to just understand a little more about the biology and how that might affect our sampling for future.
Life stages are interesting because there's all of the reproduction and release and things like that, and the biomass. So they really have like a different impact, but they can create a strong variation altogether if they are mature, large, and they reproduce. So
yeah, thank you. There's a lot of challenges.
Yeah, but that's good. That's good to know which factors is missing.
Mm-hmm.
Thank you, Jonathan.
Hiya. I'm based at the Federal University of Pará. For those that don't know, that's here at Belém at the mouth of the Amazon. So we're placed to do both freshwater and marine studies and a lot of estuarine studies as well. Our research group works on a whole range of different questions, from bulk samples to eDNA in terrestrial and aquatic environments. A lot of the time, what I find it comes down to is optimization, particularly here. I mean, our group has a reasonable amount of funding, but obviously you want to make the best use of that. So the replication that you can do, the people that are available to be in the same team, to go to the field, and all those factors tend to influence what you can carry with you, and therefore what you can do in terms of sample design as well, without having to do effective reloading. This is because obviously we have to do a big field trip. If you're near a base, then things are a lot easier. One of the other big factors that we have to consider here is human risk. We don't have the same life-saving resources that most of you guys have in Europe, North America. We also have a lot of interaction with local representatives from agencies when we go to the field. So we try to find out from them where is good to go. And we also often try to invite them into the field so that we can understand better the local sampling within the system, because they often understand where things are happening, where there may be influences. Avoiding anthropogenic influences is a big issue.
Do you have a list of questions that you're asking them, or you just like get a
It's a
feeling
fairly
on
open discussion. Usually there are themes mainly. So if we do a lot of metabar coding for metazoans in general. So one of the things that we try to avoid is anything near farm runoff, anything near strong fishing activity where the nets passing through the water are going to cause just a big cloud of eDNA. Yeah. What else? We do try to do in general a lot of replicates, because we find that here, I think as with Prisma, I was saying, you know, we're in an environment where there's a lot of biodiversity, and we want to really sort of map the biodiversity. We haven't really got much into temporal sampling yet, although we do have some places where we're beginning to do that. And more the replicates and the spatial distribution. I think it was Nick Schulte said that was very important. They're looking at that sort of thing. So we're doing those sorts of questions as well. Trying to look at plume effects and current effects on the DNA, and stratification. Obviously here at the Amazon, we have the Amazon plume, which is a little bit of an issue, with massive sediment rates also being a big problem for filter blockage. Yeah.
Yeah, I see some guidance can be like trees and where questions, and this is good to know, like, you know, risk is one that often we forget, but that's essential. So we always have the possibility that some people, even if we have the best practices, they need another option that might be safer. So when we're discussing about the best practices, we need to consider all of those limitations, you know, to have other branch, other options for the best practices. So that's very different than here.
Yeah, it's a very different system. So maybe some of the things that I can contribute to can be a little bit more of a tropical megadiversity kind of issue. They're very, very different challenges.
Yeah, that's great. Thanks. Katie, how will you suggest to wrapping up? I think some people didn't talk yet, so maybe they can add their presentation in the chat or in the notes. And maybe I was hoping we can have a discussion about missing gap and how we can communicate the best practices, but we didn't have time. So maybe they can add ideas in the notes.
Right.
But
I would say please add anything to the notes that you would like to share. I think it's been really interesting hearing about all the different types of challenges that people are sampling in. I think for me, yeah, I think all our discussion about how we see, you know, eDNA is reflecting where organisms are, but then taking into account how the physical dynamics of your system is affecting eDNA transport. I think all that's very interesting to take into account, like how to design future eDNA experiments. But yeah, thank everyone for a great discussion. I'm going to put all the notes in our session Google Doc folder, which you can get through Kiko chat platform, but let me see if I can get the link here real quick too. And I've also will put a copy of the chat in there as well, if you would like to reference that.
yeah,
Here, I think is the Google
I
Drive folder.
That's awesome. So we didn't have time also to review the questionnaire, but I will look forward to see the challenges from everyone. And hopefully we can have a next session at some point. And it has been very interesting in my found it. So thank you very much, everyone. Katie, do you have any things you want to add, or?
No, thanks everyone for an excellent week. Yeah, I really appreciate everyone's participation. It's been great chatting with you.
Yeah, it has been great. Thank you so much.
Thank you, everyone. Thank you, Anais.
Thanks.
Katie and Anais.